All right, well, welcome everyone. It's nice to see you, and I'm sure and that we'll hear, I hope, from the people who are joining us on Zoom this morning. The title of my talk is An Updated Perspective on Genome-Wide Association Studies. I do want to get into some fundamentals of the study design and methodology behind a genome-wide association study, but I'm going to wrap up the primer lecture on showcasing a modern genome-wide association study, one that was just published a couple months ago and really showcases the power of this singular tool in human genetics and genetic epidemiology. And then I'll wrap up with a very short vignette on how we can leverage the results of many different genome-wide association studies to try to understand some fundamental aspects of a particular trait or disease. So genome-wide association studies leverage a fundamental concept in genetic epidemiology called heritability. Heritability is a statistic on a disease or a trait that estimates the degree of variation in this trait in a population that is due to genetic variation between individuals in that population. There are many ways to estimate heritability. I've listed a couple of the most common techniques. We can compare individual phenotypic variation among related individuals in a population. If you have a study design of a nuclear family, for example, you know expected trait covariances between the individuals and the families based on the amount of the genetic the DNA that's been inherited through the pedigree. You can also estimate heritability by examining the association between individual phenotype and genotype data, and by modeling summary level data from genome-wide association studies. So you only really perform a genome-wide association study on a trait that is heritable, as the purpose of a genome-wide association study is to discover particular genetic variants that are associated with your outcome. And if a trait has no heritability, then perhaps you would go looking at other epidemiological methods to find the relationships between that trait and other things, other risk factors, not genetic risk factors. Another fundamental principle of genome-wide association studies is this idea of genetic architecture of human diseases and complex traits. Now, a genetic architecture of a trait is comprised of the number of genetic loci that are associated with that trait, their frequencies, if being common, low frequency, or rare, and then their effects, and the strong or weak. So this graphic that I have up here is from a 2008 review in Nature Reviews Genetics that showcases that when you have a very rare variant with high penetrance, those are the type of variants that are studied in rare disease genetics, whereas in genome-wide association studies, most of the variants that are discovered are common frequency and low effect.
May I ask a question about
absolutely.
that?
Yes, please.
In that bottom left-hand corner, are there really cases where the VAF is very low with weak effects, or is that just we're filling up the entire space with the bubble?
You mean the hard to identify genetically section? Well, back in 2008, our ability to find genetic variants that were so rare for the purpose of genome-wide association studies was very limited. So I would say that nowadays we have a much better ability to identify variants that are very rare that have low effect, because we have, in many cases, whole genome sequence association studies that are being performed. So cohorts with full spectrum of genetic variation due to the fact that the technique of whole genome sequencing has been performed on the samples. So I would, and I'll say another bit is statistical power, which I'll talk about a little bit, will play a role in being able to identify genetic variants that are very rare with low and moderate effects. So extremely large sample sizes would be necessary in that case. Are there any other questions? All right, so the purpose of a genome-wide association study on a particular trait is to discover or define the genetic architecture of that trait in the particular, with the data sets that you have available, the generated genotypes, et cetera. So we most commonly or traditionally, the genome-wide association study tests this common disease, common variant hypothesis. So the GWAS were developed for variants that were of some level of frequency, of high enough frequency in the human population to have been put on this technological product called the genotyping array. And simply, we've got single nucleotide polymorphisms, SNPs, which now have, we have the ability to read out genetic variation that is more than just a coding change, but that might span many from, to be a deletion of an allele or a variant, or be a substitution. So nowadays we have the ability to see much more sophisticated allelic variation than just single nucleotide polymorphisms. But generally, we have an allele, a place in the genome where there is a change, and the allele has a kind of a observed frequency in the sample and the population that you're studying. And our hypothesis is asking the question, is the presence of this SNP allele associated or correlated with our disease and trait? So C allele being perhaps the example that I have circled here with 25% frequency in this certain population. We want to know if the C allele is associated with the trait. We generally want to just scan the genome, and at every allele where there's a variant observed, ask this question. So we're going to be performing a statistical test at millions and millions of places along the genome. But if you're going to be performing a statistical analysis, you have to put together a model of the variables in your association analysis, and that requires us as the analysts, studying this question, to decide how we're going to treat this genetic, the genotypes and the alleles observed at this particular location. How are we going to code them for our numerical analysis so that as we look for a correlation? I'm putting up here the 3 common genetic models and depiction of what we are imposing in terms of what we're looking for in this statistical analysis. The additive model is one where at this particular location, you're looking for a correlation with or an effect of the C allele. For every individual, you'll be counting how many C alleles they have in their genotype. So an individual in our analysis has either a 0, 1, or 2 as the variable value that's used in our analysis. You could also model a dominant or a recessive effect. Nowadays, there are statistical tools that allow you to do GWAS with more sophisticated models like a dominance model or a recessive model. This is one of the things where after 15, 20 years of doing GWAS, I just take for granted. But when Genome-Wide Association first started, there was a question about what was the best coding of an allele if you're doing a scan of millions of alleles across the human genome. And there was some literature that came out that concluded that if you don't know what the model is, then the additive model will be the best, generalized model to use in the set, for these millions of variants. Any questions about this? I did build a bit of time here for us to have questions. I think if you have a question, you should go to the microphone or get it be passed along to you. Thank you. Yep.
In the graph in the bottom left, what's the y-axis of the trait?
Think of like a regression analysis where you're trying to find a best fit in a relationship between a covariate and your outcome. So if we were going to that this line could represent the expected change in the trait value between people have 0 or 1 or 1 and 2 as their kind of value for the genotype or for the allele. Yeah, so I would say that this is maybe like an idealized model, whereas for a real analysis, you'd have a scatter of lots of different points, and you're trying to do a best fit, like just a regression type analysis. Is there a question from online?
Yeah, we had one online. If you could go back to the figure with the effect size and the allele frequency, that bottom left corner Nicole was asking about.
Yes, yep.
They're asking that the size of that bubble, they're saying is still probably a pretty big bubble, but we don't have the heterogeneity across populations to be able to capture those variants.
Yeah, so yes, the point is that low frequency variants that have slow effects are still going to be hard to identify because I'm looking at the right population or looking across many populations, heterogeneity in the fact that the allele may not exist in some human populations. And so there's lots of sources of heterogeneity, I would say, that would contribute to these variants being hard to explore. So that sounded less like a question, but maybe a comment. I appreciate that. And people who are on the Zoom, you can feel free to fill in and more comments, because I'm going to keep things at a pretty high level during this primer. Go ahead.
In which case would you use a dominant or a recessive model over like an additive model? So like in what context is that better?
I would say if you want to do a genetic discovery analysis and you want to explore you want to test the possibility of there being genetic alleles with a recessive effect across the genome. In the field of type 2 diabetes, we recently had a paper published showing the results of a recessive model, GWAS, a very large one. And so that was quite interesting for us to find the genetic alleles that show a recessive effect, but not an additive effect. Yeah, so it's just another technique in terms of the discovery of genetic variations and exploring the genetic architecture of a trait.
Thanks.
So I think if you have a hypothesis that things could be that there could be genetic variants that have a recessive model or dominant model, or you want to explore, you would just employ one of those statistical tools that model a recessive effect in GWAS. All right, so implementing a GWAS statistical tests here. I think it's really important that if you're about to embark on performing a genome-wide association study, or trying to understand a genome-wide association study, that you find a good textbook that starts with some fundamentals in biostatistics and start understanding the distribution of your outcome and covariates and the correct statistical modeling. I would say any genome-wide association study starts apart from the genetics, looking at your outcome, covariates, confounding, making sure that you understand the outcome that you're studying and are using the appropriate covariates to control for confounding in your model. I'm not going to talk too much more about this, but there's tons of literature out there about using genetic principal components as a method to control for the problem of population structure. And this the problem is generally stated as there being differences in allele frequencies between groups in your analysis sample that are correlated with the differences in the trait or disease that you're studying. So before you employ a GWAS, study the associations between your outcomes and your covariates and understand the structure of your sample so that you are starting off with a good epidemiological model before you add in the genetic variants from the GWAS. So, yep, the basic question, I'm just repeating it here, are the alleles or the genotypes of the SNP correlated with the phenotype? So for quantitative traits, here are some examples: height, blood pressure measurements, gene expression levels, other physiological measures. So within a cohort, you have these variables available to you. We set up a simple regression framework, and most all modern genome-wide association study methods or tools and techniques just extend this model. And there are edge cases and there are some pretty cool methods that are out there, but for the purpose of this primer, I want us all to just become familiar with the basic model that's being explored here. So again, if you're not familiar with a linear regression or multiple linear regression, and you want to become more familiar with it, I would have you go back to some basic biostatistics textbooks and just start learning about trait variability, correlation, and then regression, and then multiple regression. So we're going to fit a regression, best fit line like that between a trait and a SNP, controlling for your confounders. And if there is a relationship, the regression estimate, which here I have represented by this beta SNP, will be non-zero. Generally, if there's, we would not expect the regression estimate to be non-zero if there is no association. So under the null hypothesis, which is a statistical term for no effect, our estimate will have the estimates of those variants under null hypothesis would be zero. Of course, when you're sampling, they're not, which is what we're going to get into in a couple minutes about false positives. But for a SNP that is associated with your trait, when your regression estimate is greater than zero, then you interpret that as saying the allele is associated with higher values of the trait. And when the estimate is less than zero, your allele is associated with, I have a typo, lower values of the trait. Now, if you have a case control study design or a binary trait, the question becomes if there are different in allele frequencies between cases and controls. And then the previous regression framework is extended into a logistic regression where the effect estimate is an odds ratio, which is a measure of the increased or decreased risk of this disease on a multiplicative scale. And the odds ratio under where there's no effect would it be expected to be 1. So an odds ratio greater than 1, it means that there's increased risk of disease associated with the allele. An odds ratio less than 1, decreased risk of disease associated with the allele. When you perform a statistical test, you're going to have a p-value as a result of the statistical test. The definition of a p-value is a probability, so it's a chance of observing as strong of an effect as the one you have in front of you for that particular variant if there is no association. And so getting into some more statistical language, so if this is unfamiliar to you, then I would, again, invite you to go and explore some of the fundamentals of biostatistics. But when we're setting up a statistical experiment, we set prior to doing our experiment kind of a false positive rate that we would expect to, that we're going to use. And you'll hear sometimes people saying a false positive rate or an alpha level of 0.05 as being one that's an acceptable false positive rate for a single experiment. And this means that if there is no association between your trait and your outcome, if you were to go, let's say, and sample, create samples in your population, many different samples of this particular variant, so maybe perform any GWAS in many different groups of the people, then about 5% of these tests would have a p-value less than 0.05. So just due to sampling variability and just randomness, you're going to get some results that have p-value less than your threshold of determining something is important. But the problem with genome-wide association studies is that when multiple tests are performed, the probability of having at least one false positive is greater than alpha. It's just like a numerical fact if you work it all out. So we're not doing a test of a single genetic variant. We're doing a test of millions or hundreds of millions of genetic variants in this one sample that you have for your genome-wide association study. So to maintain an overall false positive rate of 0.05, you have to adjust this threshold that you're going to decide is sufficient for you to call something statistically significant. And it's based on the number of independent tests being performed. So we have this Bonferroni method for correcting p-values or adjusting a family-wise error rates in an experiment where you're looking at more than just one single statistical test, where you take your overall false positive rate and divide it by the number of tests. Now, there's a lot of early literature around on what should this p-value be in the case of genome-wide association studies. And the field settled on this number of 5 times 10 to the minus 8, which can be obtained in a couple different ways, but based on the genotyping array technology available at that time, and correlation, linkage to equilibrium, which is another population genetics term that exists in the human, in the most likely sample that were being studying at that time. There were about 1 million independent regions of the genome. So 0.05 divided by 1 million, you get something around 5 times 10 to the minus 8. So this is one of the origin stories of that significance threshold for genome-wide association studies, which is commonly used these days. More modern, more recently, there have been other methods of obtaining statistical significance thresholds, and sometimes for a genome-wide association study, you'll see something like around 10 to the minus 9 or 10 to the minus 11, 10 to the minus 12, all justified by trying to estimate the number of independent tests being performed in a large-scale genome-wide association study.
We have a quick question in mind. Could we calculate the risk ratio instead of the odds ratio?
That's a great question. So going back to the model for a case control study, I would say that sometimes a risk ratio could be a more appropriate measure of association. But since we're using a regression framework, perhaps it's easier to obtain an odds ratio from a regression. The way that this whole formula works out, and you want to get to a measure of risk in the case of a binary outcome, you take that the regression coefficient for the SNP, the beta, and use it as it's e to that beta equals an odds ratio just mathematically. So I would say that in genome-wide association studies, we use odds ratio because of the ease of calculating it from the regression, a logistic regression. It's a good point because if you're studying common diseases in epidemiology, you learn that perhaps an odds ratio is not going to be a good approximation for a risk ratio. So it's a good point to keep in mind. We have another question. You can use the microphone.
Yeah, from the previous slide, when you have the regression equation set up,
yeah,
So is the goal of the study to discover like these beta values? So would that be done through a grid search, or is it just the beta for the SNP?
So the solution to, I would say that it's a standard solution for solving like linear regression, which is not a grid search. There's actually a numerical solution.
Oh, okay.
So it's not a machine learning approach. It's just kind of there's a set of formulas that would be used to obtain the estimate from the sample. And then the many extensions that go off from this, it gets more complicated, but that's the gist of it. It's basically calculated from the observations in your sample. There's a lot of literature if you want to learn more about how the regression estimates are obtained in a multiple linear setting.
Okay. And is it like we're finding all of these betas or just the...
Yep, you actually fit the whole model for every SNP. And sometimes if your model is really complicated, and this is why we have to use more modern techniques, it can take several seconds or like half a second to actually get these numbers from your the data that you have. And so when you're doing 1 million or 100 million or 500 million SNPs, it can take a really long time. So there are now a number of techniques that speed up these calculations through estimations, but I really am not going to get into that for the primer.
Thank you.
Oh, sure. All right, so let's get into some of these other. Okay, we move quickly through some of these. So I talked about the genome-wide significance threshold traditionally being 5 times 10 minus 8. I'm going to get into describing, well, here, this is a depiction of a Manhattan plot, which we often see for genome-wide association studies, where every point is a SNP that's tested. And on the y-axis, we transform the p-value of that SNP by taking the negative value of log 10 of that p. And there's no reason why it's this and not something else. It's just that we want a way to let variants that have low p-values be, seen easily. So this transformation was applied and has worked really well for us. So you have, this is early type 2 diabetes, GWAS. You have a region on chromosome 10, where you see a number of genetic variants with low p-values. These are all in the same locus. They're correlated with linkage disequilibrium in that area, which is, I think I said earlier, a population genetics term. But you can see how the places of statistical significance rise above the rest, and they look like the skyline of Manhattan. We use quantile plots or QQ plots to look at the results of a genome-wide association studies to make sure that the underlying model is good and that confounding is well controlled. This is because that under the null hypothesis, there is we expect so when there are no associations between a variant and your outcome, we expect the points in a quantile plot to follow the x equals y line here, just the way we've constructed it. I would say nowadays we have very well-powered genome-wide association studies. So this was from the early days when we did not have well-powered, and by powered I mean statistical power or ability to detect associations due to having a large enough sample size, which I might get into a little bit. Finally, the genome, well, not finally, this is kind of wrapping up some of the primer, like the really basic terms that we use. The genetic variants that we're studying, we don't really know if they are the ones that are causing the association between causing whatever I guess biological or physiological change that impacts your outcome or your phenotype. And back in the early days of genome-wide association studies, we were using genotyping arrays, which were pre-made products that read out the genotype of a particular individual at all these different set locations. And so maybe was more like this, the situation where the red places along this strand of DNA were what we actually had available to us in our genotyping array data. But the true association here, which is kind of this blue bar or dot figure thing, was the causal variant, but it wasn't included in our genotyping array. Thanks to a technique called imputation, where we can leverage sequencing data in populations to kind of fill in the gap, because humans share this, very, very, very high percentage of their genetic sequence. We didn't. So before we had this imputation techniques available to us, we just had what was available in genotyping arrays, which meant that most likely we were not observing any causal variants, or the causal variants at a location. And the reason it worked is because of this linkage disequilibrium, which is the correlation between observed alleles that are just present due to the changes of allele frequencies in human populations.
We have a quick question on the previous slide. Yeah. Could you touch on inflation in the lambda, looking at the QQ plot?
I would. So.
And how to interpret?
Well, I think that may be a little more than I want to get into during the primer, but I would say that if you're studying in the context of genome-wide association studies, if you look at you can study the distribution of all the variants in this way, and you can kind of measure it using a metric called the genomic control measure, which is the median test statistic producing that would be somewhere in this distribution. You compare the observed median test statistic to the expected median test statistic, and that basically says, down in this part of the distribution, where you find the median, that the observed median is larger or smaller than what you would expect, and that's just a key. That's a indication that there could be some modeling issues, some undetected confounding in your analysis, and that you should go back and revisit often. So traditionally, it's been population structure, the need for controlling for a difference in allele frequencies and subpopulations in your sample that could be corrected through the use of genetic principal components, but there are other reasons that could change, that could lead you to do a different type of modeling. All right, so we have depicted here a regional plot, which is a zoomed in version of the Manhattan plot. And for most genome-wide association studies, there'll be loci where there's an association. The authors of the study want to say something about those regions, so they'll use a kind of a regional plot that depicts the linkage disequilibrium statistics between your here I'll use my mouse, this is the index variant, this is the variant that has the lowest p-value or that you are the most interested in, and you can kind of calculate pairwise linkage disequilibrium with all the other variants and then put them on a plot. So it's a useful type of depiction. Next primer, if you're going to be studying, learning about eQTLs, the idea of things at the regional level will become really important. Finally in this whole, we're gonna go into a couple different themes next. I wanted to contrast the observed genetic architecture of common diseases and biomedical traits. This is from a review in Nature Review Genetics from 2018 called Genetic Architecture, the Shape of the Genetic Contribution to Human Traits and Diseases. So on the left-hand plot, I'm showing you I'm sharing the genetic architecture of type 1 diabetes versus type 2 diabetes from known at that time. And you'll see that there are quite a number of differences in the genetic architecture. You'll see that there are a number of low frequency variants that are associated with type 1 diabetes. There's this point up here, which is a frequent variant that has a very strong effect on type 1 diabetes. Contrast that with a number of low effect variants across the allele frequency spectrum. I would say that as the statistical power of studies have increased, we actually have been able to fill in a lot of the variants in this area for type 2 diabetes. On the right-hand side, we have vitamin D levels and LDL cholesterol. Here showcasing that vitamin D level, maybe it's a less heritable trait. There are fewer variants associated with vitamin D level. I'm used in this particular figure, and a number of, and many more associated with LDL cholesterol. So a trait that you're studying could have any sort of genetic architecture. You might have a hypothesis about what the genetic architecture is, but I would say the purpose of further genome-wide association studies on all these different traits and outcomes is to really kind of define the genetic architecture of that trait. Are there any questions? All right, so I'm going to dive into some examples. Back in 2010, there became this sort of celebratory depiction of all the genome-wide association studies that were discovered, because it was a little slow going in the beginning because we didn't realize that the statistical power could be as well. We didn't realize that the effects that we would see, I'll come back here, were going to be so low, and that the ability to detect these in a genome-wide association study depended on the size, the sample size. So back in 2010, great, we're celebrating. We found some genetic variants that were associated with many different types of traits. In 2014, even better, this is good, we're gaining momentum. And in 2019, it's saturated. So many different groups across the world are doing genome-wide association studies on so many different traits. Meta-analyses are coming, are happening to combine as many small samples into a big meta sample. And by this time in 2019, it was pretty well established that genome-wide association studies were successful and were a good tool to find the genetic variants associated with traits and outcomes. I would say it was hairy at the beginning. Around 2010, there was a lot of commentary that there was a lot of money being spent on these genome-wide association studies, and nothing was really coming out of them. The small effects are still maybe the norm. So a lot of the things that flow downstream from genome-wide association studies are now built to take advantage or use these small effect genetic variants that are found in many different ways. All right, so now I'm going to get into an example of a modern genome-wide association study. This is a publication that just came out 2 or 3 months ago from the VA Million Veteran Program. And this is a group, I know many of these investigators well, and so I was really happy to see when this big paper came out. It's a single cohort, whereas often, like I said, these meta-analyses will be performed to combine many different cohorts into a single meta-analysis. But here we have a depiction of 2,068 traits being studied in the Million Veteran Program. And this particular analysis what is important because the entire spectrum of diversity within the United States, I would say, could be better represented in the sample from the Veterans Program. So Overall, the number of people contributing to this analysis are 635,969. It's just so cool to see this. Their particular, the traits that they're trying to use, the heritable traits that they're using in this analysis had to do with fee codes, which are just medical record codes, or outcomes or diseases that are derived or obtained from medical record data. They have laboratory measures, vital statistics, and then survey questions. So the type of variable, or the type of variable outcomes being studied here can vary. And this is a good example of the type of data you can use as outcomes in a genome-wide association study. Let's see, this is a kind of a depiction of the traits and the frequency of the binary traits on the left, and the sample sizes available for the quantitative traits on the right. And so here, I guess I would flag that even in a single sample, studying a trait, you're not going to have the same sample size for every trait because you are subject to data availability. But it was really cool to see how there's more than 1,000 or 10,000 or 100,000 individuals with a particular condition being studied here. So we have the Manhattan plots of their binary trait results and then their quantitative trait results. And then the on the right-hand side is the genetic architecture plot, where they've taken the minor allele. So for any variant in the genome, the minor allele is defined as the less frequent allele, and then plotted the effect, either the beta in the case of the, I've flipped these because I have beta up there and then odds ratio down here. So the odds ratio goes up with the binary traits. But the point being is that it shows how you can find common and rare variants with large effects on the either the outcome on the odds ratio scale or a trait on a kind of probably some normalized scale here. To put all the traits in the same plot, you'd have to normalize the scale or the unit of the betas. So, I mean, you can see here that variants in that hard to detect region are here. And that really showcases the power of a genome-wide association study. Okay, I'm going to wrap up with one more example. All right, so this is our toolkit, and I've gone over most of all of these. I have a little plug for some good resources that people could use that are up to date. The very small vignette is an analysis run by a postdoctoral fellow in my lab, or actually she's now been promoted to as an instructor. So Magdalena Sevilla Gonzalez ran an analysis to detangle the heterogeneity of hyperinsulinemia. So this is fasting insulin levels, along with my colleague Miriam Udler, who's also an associate member here at the Broad Institute. And here, I'm just gonna go skip through some of the basics, but our aim here was to use the genetic approach to elucidate or explain the mechanisms that underlie the relationship between insulin concentrations and cardiovascular risk. So the gist of it is that there are many reasons for a fasting insulin to be increased in a person, and we want to understand what those reasons are. And we took an approach where we found a set of variants associated with fasting insulin. We leveraged the Type 2 Diabetes Knowledge Portal, which is a wonderful resource that came out of the Broad Institute and is now a worldwide success for curating the results of genetic analysis. But then we took traits that were from all these other, or we took GWAS results from all these other traits. And we're going to leverage this concept of pleiotropy to find similarities in associations. So this is just a depiction of a heat map showing the effect of these. We know these SNPs are all associated with type 2 with fasting insulin, but now we're looking at what the associations are with all these other traits, and we see a place, something that's kind of different. What I'm highlighting here is a particular variant which has an association with triglycerides level and a bunch of other traits, but in an unexpected pattern. So most all the other SNPs, if they're associated with triglycerides, might have been associated with increased triglycerides, but this particular SNP is associated with decreased triglycerides. And pleiotropy is defined as an observation of a particular variant being associated with many different traits. And then there's a whole field of trying to understand how that works, if it's one trait affecting many traits, or if the variant through different mechanisms are associated with these different traits. Here, our hypothesis is that there may be a common mechanism. So another set of variants we found having some pattern of associations across many different traits. We found that very interesting. And so we're using this as a starting point for a more in-depth analysis. All right. So I'm going to stop here and we have time for a few questions. And I think then we wrap up around 9:25, between 9:30, and then people can go over to the MPG lecture.
Hi, thank you so much. I was wondering, is it possible to bring a GWAS approach to bear on larger mutational events like structural variants? And if so, how would you do that?
So that is an extension of genome-wide association studies where you would want to have a cohort or multiple cohorts where structural variants were the alleles that were being studied. So structural variants are defined as insertions or deletions in the human genome that are greater than 50 base pairs long. So as I was mentioning earlier in this talk, that we have now the ability to see these insertion and deletions as alleles in our sequence. There's also now some techniques getting better every year. I think this is an area of really rapid development in human genetics to look for large-scale genetic variation. And so in the case that you have a data set where you have structural variant calls in a cohort, you can perform a genome-wide association study just like that I've shown here. So you're just starting with a different sort of alleles. But the real challenge in the field right now is getting these structural variants that created using the available genetic data that you have in a sample. And there are a lot of nuances about that having to do with the technique that we use typically to get from a person's DNA sample. We cut it up into tiny little pieces, and then there's a process through which the genotypes are read out, and so that process of cutting up the DNA interferes with the detection of large-scale changes. So at a very small scale, there's these long read sequencing techniques that are being employed, and hopefully there'll be some new methods and new approaches that will allow many different cohorts that have genotyping data to leverage kind of maybe a reference panel of this long read sequencing to kind of impute the long read type structural variants in their cohorts. So just an exciting extension to genome-wide association studies.
Hello. So one term that I see often in GWAS literature is the liability scale. I wonder if you could comment on the idea behind converting a trait value to the liability scale.
I think I thought about putting something like this in the heritability piece because you can technically only calculate heritability of a binary trait on a liability scale. I think it's slightly more advanced concept, but a liability the concept behind liability assumes that there's some underlying distribution above which, when a threshold, a person's value for that whatever underlying trait passes some threshold, then they're going to be diseased, or there's a pathologic, like there's a consequence. And so there's a lot of statistical work to explain this. So I'm not going to do a very good job on the fly. The books that I actually have used as references have a much better detailed explanation. There's more to be said about, and it really has to do with the underlying model, like the assumptions that between of how you get to this disease status and how you can measure the underlying causes, which are typically unknown.